let me just say a word about integrity. I regret having to say this, but I have to say it every year. I value integrity really, really highly. You guys, you're MIT students, and I shouldn't even have to bring this up. And 99% of you are absolutely outstanding. Okay? But there's about 1% of students, I think, at MIT who've decided that it's okay to cheat. And I just want you guys to know that if we catch somebody cheating in this course, and we have some built-in controls to monitor that, then I'm going to use every tool at my disposal and every ounce of energy I have to get that person dismissed from MIT. Because cheaters do a disservice to all of you honest people out there, right? And worse, they then go on to do horrible things in the world, as we've seen from financial crises and our Congress and all the other problems that we have. So it's really important that you have integrity. I'll tell you, people can take everything in the world that you possess away from you except your integrity. They can take all your money, they can take your freedom, but they can't take your integrity. So you should preserve it. All right, enough harsh talk. Like so many great accomplishments in human history, the origins of biochemistry are rooted in the production of alcohol and alcoholic beverages. How many people here have read the book, A History of the World in 6 Glasses? Oh my God, you guys got to read this during spring break. History of the World in 6 Glasses. Because 2 of those 6 glasses, maybe it's 3, are alcohol. Alcohol has been a dominant force in human evolution and science and technology since the dawn of time. And our knowledge of alcoholic beverages dates back at least 3,000 years or more. Okay? And all of our understanding of alcohol and alcoholic beverages are really based on one specific reaction, and that's this reaction. It's the reaction of C6H12O6, or glucose, into 2 molecules of ethanol and 2 molecules of CO2. Now, these proportions of ethanol and CO2 to glucose were actually worked out, believe it or not, by Lavoisier. And the process that converts glucose into ethanol and CO2 is catalyzed, of course, by yeast. This is something that the Greeks knew about, and the Greeks had a special word for yeast. They called yeast zyme, Z-Y-M-E. And the Greeks knew that zyme was required in order to produce gas in bread, and it was required to turn grapes into wine. And the role of yeast in catalyzing this reaction was further elaborated by Louis Pasteur in the 1850s, who showed that you could only get this process to go, at least under the conditions that he explored, if yeast were present. And this led to 2 schools of thought about the process by which glucose could be converted into alcohol. And those 2 schools of thought, one of them was called vitalism, and one of them was called mechanism. Anybody heard of vitalism before? So vitalism is good. So vitalism was this idea that fermentation was the result of some kind of magical life force, something that was present in living cells that was governed by entirely different laws than the laws that apply to inanimate matter, right? That this was somehow self-determining or something special, right? This was the life force. Okay? And mechanism, of course, was exactly the opposite. Mechanism simply said that natural phenomena are based on the same fundamental laws, right, of chemistry and physics that everything else applies to. And the question was the process by which glucose was converted into ethanol and CO2 some magic life process, or was it just the same old chemistry and physics that we all knew about? Now, Pasteur was determined to solve this problem, and so he took yeast and he took glass and he ground up the yeast and he made a cell-free extract of yeast. And he found that cell-free extract was completely unable to catalyze the conversion of glucose into ethanol. Clearly, vitalism must be right? There has to be some magical life force. Now, we all laugh at this. We snicker at this, and we think, aha. But I can tell you, there's plenty of stuff in the lay literature that talks about vitalism. They may not call it vitalism, and they may not write it this way, but they use all kinds of other explanations that claim that there's something magical about life that we can't somehow explain with physics and chemistry. Now, the change came, the real breakthrough here came in 1897. The change came in 1897 by a German chemist whose name was Eduard Buchner. Not the same Buchner as the Buchner funnel, but the name is the same. And what he did was he took yeast and he mixed them together with quartz and kieselguhr, which is really diatomaceous earth. And in this, he used a solution that had 40% sucrose. And he, in fact, was able to make a cell-free extract, something that had no yeast in it. And that cell-free extract was able to convert glucose into ethanol. Okay? And this was really a landmark finding. And what Buchner said was that there was something in the yeast that could do this, and he called that magic property, he called them ferments of zyme. Something in zyme, in yeast, that was able to ferment glucose into CO2. And we now know what this stuff is, right? That these days our modern term for this is an enzyme. But that's where the term enzyme comes from. Okay? But this actually was the birth of biochemistry in 1897, and that really opened the door to the scientific study of reactions in living matter. Okay. So with that as a background, let's talk a little bit about where biochemistry fits with the other subjects. So biochemistry, the thing that's going to explain these properties of living matter is, I would argue, is a great unifier of biology with chemistry, physics, and math. And so biochemistry, if I write it here in the center, receives, you could think of this as receiving inputs or able to explain a variety of other areas of biology with which you might be familiar. So genetics gives us inputs, not observations from cell biology give us some inputs, and observations from what we now call molecular biology all give us observations that we then in turn need to be able to explain biochemically. We can also use structural biology, that is looking at the structure of macromolecules, and biophysics to get inputs into biochemistry. And what emerges, of course, when we do this, what our goal is in this course is not to simply describe some genetic interaction or to point to some structure inside a cell or to tell you with a bunch of cartoons that represent molecules what's happening. It's really to get at mechanism. It's only by understanding the chemical mechanisms that are responsible for the things that we observe in cell biology, genetics, molecular biology, and so forth, that we really can understand things. And then, of course, the beauty of this is the output that comes from understanding biochemistry, and that's understanding anatomy and physiology. Because now we can understand, for example, things like how the heart pumps and how the lungs manage to exchange oxygen and how fluid flows through your kidneys, because we understand the biochemical mechanism that underlies all of this. So if we say that biochemistry is the chemistry of the molecules of life, if this is the biochemistry of the molecules of life, it's, of course, as you know, carbon-based. So why carbon? Well, there are only 5 elements that can make greater than 3 bonds, right? Otherwise, you end up with like long chains of atoms, and that's not what we need. We need to have 3 or more chemical bonds. And as I said, there are only 5 elements that can make greater than or equal to 3 bonds. And those elements are boron, carbon, nitrogen, silicon, and phosphorus. So why don't we have, for example, other types of things? Well, metals, of course, as you know, the reason that we don't have life with metals is because they really form ions and not so much covalent bonds. They also form metallic bonds that we'll talk about later, right? And then, of course, noble gases, which are non-reactive. So that rules this out, that rules this out. We're stuck with these 5. Now, why not have a life form that's based on boron? Right? Well, boron, as you know, has 3 valence electrons. Okay. And this electron, this state of electron deficiency, right, limits, it's electron deficient, and it limits its stability. How about nitrogen? This has 5 valence electrons, okay? And this is really electron rich. But all of those electrons make for repulsive forces that limit the extent to which we can make long chains. What's a great example, right? The problem is I can make short chains. I can make things like NNN, right? But what's the problem with molecules that contain a bunch of nitrogens all stuck together? Anybody know? Yes? They become explosive, exactly. Okay? These kind of nitrogen compounds are great for rocket fuel, but they're not great for life. Okay. Carbon, as you know, 4 valence electrons, very stable, works really great. What about silicon? Right? We know that silicon, right, it's right below carbon in the periodic table. Right? So what's the problem with silicon? There was a great Star Trek adventure many years ago about silicon-based life forms. They were like rocks, right? Why don't we have a silicon-based life form? Well, the problem with silicon is that the radius is too large. And as a result, we get limited orbital overlap. And so as a result, although silicon can form networks, a lot of the time silicon basically ends up forming weak silicon-silicon bonds. Okay? So when you have a silicon-silicon bond, it's a weak bond. How about phosphorus, which is below nitrogen in the periodic table? It's even less stable. We have to talk about 2 types of bonds. We have to talk about homonuclear bonds, which are, of course, carbon-carbon bonds, and heteronuclear bonds. Things like carbon-nitrogen bonds, carbon-oxygen bonds, right? Esters and ethers, carbon, oxygen, phosphate. I'm not drawing all the bonds, but just to give you some idea of what I mean by heteronuclear bonds. Now, these in general are less stable than homonuclear carbon-carbon bonds. And as a result, it's easier to have reactions that utilize the heteronuclear bonds. And so these are commonly involved in biochemical reactions. Now, our decision to say that we believe in mechanism rather than vitalism, and our understanding that it's these heteronuclear bonds that are going to be targeted in biochemistry, means that the very same rules that you learned in 512 in organic chemistry are what we're going to apply when we try to understand biochemistry. But this introduces one complicated aspect. So we can say that it's the same reactions as in OChem. But, this is a really important but, they're applied under physiological conditions. What do I mean by physiological conditions? Well, I mean these have to occur under 1 atmosphere pressure. Okay? You can't, it's not like in the OChem lab where you can put it in a pressure reactor and jack the pressure up to, 37 atmospheres of nitrogen. Can't do it. Got to do it in 1 atmosphere. You got to do it at 37 degrees C, right? Or something similar, right? I can't put this in refluxing bone dry acetonitrile in a condenser apparatus at 90 degrees for 7 hours. Can't do it, right? Got to do it at 37 degrees. Furthermore, I have to do the reaction in 55 molar H2O. Now, I have 2 comments here. First, I'm from Baltimore. The way I say H2O is water. Many of you say it the way my wife does, which I think is water. I can't say it her way. So when I say water, you know what I'm talking about, right? If you're from Philly, you say it the same way. 55 molar water. I can't do this in benzene. I can't do my reactions in toluene. I have to do them in 55 molar water. And then the last aspect of biochemistry that I really want to stress that's super important is it's enantiomer specific. And what do I mean by enantiomer specific? What I mean is if we consider a common biochemical reaction, a reaction that we're going to talk about in great detail over the course of the semester, we're talking about the breakdown of glucose in muscle, and what we end up with here is pyruvate or pyruvic acid, and this gets converted by an enzyme known as lactate dehydrogenase. This detail doesn't matter. We're going to cover this in great detail later. What we end up with when we do this is this molecule. Okay? Not. Okay? I only get this enantiomer, L-S lactate. Okay? This is what accumulates in your muscle when you do too much exercise and makes you hurt. Okay? So it's enantiomer specific. Now, this is important because if I were to give you pyruvate and tell you I want you to reduce this ketone to an alcohol in the lab, it would be very challenging if I said I want you to do that reduction and I only want you to give me the L isomer. Huh? You know, if you do this in most ways, you're going to get a mixture of the L and D isomers. And I'm telling you, I only want this one. So there's something very unique about how biochemistry catalyzes these reactions, and it has to often do these kinds of reactions in clever and non-obvious ways. And all of these reactions, interestingly enough, as I said, have to be done in aqueous solution. They have to be done in 55 molar water under carefully controlled conditions, particularly pH. And so if we're going to study these kind of reactions, the ability of a particular enzyme to convert pyruvate to lactate, we have to do it under conditions where we can control the pH of the solution. So we're going to start out with the Bronsted Lowry definition of an acid and a base. Bronsted was in Denmark. Lowry was in England. They formulated this idea in 1923. And what they said was, as you know from high school chemistry, that an acid was a proton donor. And they said that a base was a proton acceptor. And that's a reasonable definition for some of what we're going to talk about. But there was an alternative definition that was provided by Gilbert Lewis. Gilbert Lewis, who was an MIT professor, he was the founder of Lewis structures. You guys all know about Lewis structures, right, where you draw the electrons around. So Gilbert Lewis came up with an alternative definition at about the same time. Okay? And so Lewis's definition, which will be much more useful when we talk about catalysis, defined what we're going to call a Lewis acid as something which could act as an electron pair acceptor. And he defined what we're going to call a Lewis base as an electron pair donor. Interestingly, just out of curiosity, Lewis is probably one of the greatest chemists of all time, in my opinion. Not only did he come up with a Lewis dot structure and the Lewis concept of acids and bases, he also reformulated thermodynamics. He coined the term photon. He had a very long and distinguished professional career. He was nominated for the Nobel Prize 35 times. Okay? He never won it because he had an enemy on the committee who nixed his name 35 times. But brilliant, absolutely brilliant. One of the most brilliant chemists of all times. Okay. Now, let's review very quickly a little bit about acids and bases. Okay. So let me remind you that all acids have conjugate bases. So if I consider any old acid, HA, this dissociates into a proton plus A minus, and this is the conjugate base. This, of course, is the acid. Now, obviously, it's no surprise, then, that bases have conjugate acids. I could equally well write that some base plus a proton gives rise to BH. I'll make it plus in this case. Okay, and this, of course, is my base. And this is my conjugate acid. Okay, and you can see I can use either definition. Clearly, the base acted as a proton acceptor, right? So it meets the Bronsted-Lowry definition. But it also donated this electron pair, and so it met the criteria for a Lewis base. Okay. Now, we would all agree, right, that vinegar is probably an acid and bicarbonate is a base. But if every acid has a conjugate base and every base has a conjugate acid, how do we decide if something's an acid or a base? And really, of course, as you know, it's determined by the extent of dissociation of the proton, if we're talking in a Bronsted-Lowry way, right? So strong acids have weak conjugate bases. And by the same token, a weak acid has a strong conjugate base. Let me say it again so you can write it down. Strong acids have weak conjugate bases. And weak acids have strong conjugate bases. Now, strong acids you are familiar with, and those are things like mineral acids. So when we talk about a strong acid, a good example might be hydrochloric acid, for instance, or hydrobromic acid, or nitric acid, sulfuric acid, etc. And if we think about hydrochloric acid, the reason is because hydrochloric acid, as you know, dissociates almost completely into a proton and a chloride anion. Okay? Whereas weak acids only dissociate partially. I'm going to write one more thing up here. A weak acid. And a good example of a weak acid is acetic acid, okay? And that's CH3COOH. And for this only dissociates a little bit into the acetate anion plus a proton. Acetate, acetic acid. And you can see I'm intentionally drawing the arrows to indicate which direction the equilibrium for this lies in. Okay? Now I'd like you to notice that there's nothing here necessarily about charge. Okay? In acetic acid, the acid is neutral, right? And the base is negatively charged, right? But I could just as easily consider, for example, I could consider ammonium, NH4 plus, right, which dissociates into ammonia plus a proton. And in this case, the acid is positively charged and the base is neutral. So don't confuse charge with something being an acid or a base. So we're going to start with the universal solvent, the molecule that all of these reactions have to take place in, and that's H2O. And as you know, I'm sure, all biochemical reactions and all of the reactions that we're going to talk about are going to take place dissolved in water. And so for H2O, as you are all infinitely well aware, we know that H2O, I'm just going to write here the concentration of water, right, is in some equilibrium with a proton and a hydroxide anion, right, where the hydroxide anion is, of course, the conjugate base, right? And we can simply define an equilibrium constant for this, which is equal to simply the equilibrium constant as traditionally reflected, concentration of H2O. Now, what does this mean, the concentration of H2O? That, of course, is just how many moles of water there are in 1 liter of water, which is simply equal to 1,000 grams, because that's how much mass there is in a liter of water, and divided by the molecular weight of water, which is 18 grams per mole. And so this is going to be 55.5 molar. That's why I said all biochemical reactions have to occur in 55 molar H2O. And so we know what this equilibrium constant is, right? We know that this equilibrium constant is equal to 1.8 times 10 to the minus 16 molar. That means that there's not very much dissociation. And so we can now define a new constant, which we'll call Kw, which is just Keq times the concentration of H2O. That's simply 1.8 times 10 to the minus 16 molar times 55.5 molar. And if we do that, that turns out to be equal to, of course, 1.00 times 10 to the minus 14 molar squared. And this must be equal to, of course, the concentration of protons times the concentration of hydroxide anions. Okay? Now, because, of course, the concentration of protons and the concentration of hydroxide anions are equal from the dissociation of water, right? That is, we know that H plus has to equal OH minus. We simply have to take the square root of this in order to find, of course, that the concentration of protons in 1 liter of water is equal to 1 times 10 to the minus 7 molar, right? And because this is cumbersome, we will take the logarithm to the base 10 minus the logarithm to the base 10 of the concentration of protons, and that, as you know, is simply equal to the pH, and that implies that our pH for 1 liter of water is exactly 7.0. Okay? Now that we know this, we want to calculate the pH of different solutions. And that's going to be important because if we're going to do biochemical reactions, we have to control the pH and we have to figure out how we're going to do that. And in order to get to buffers, I have to just review with you how we do this, okay? So, for 2 cases, for strong acids and bases. For strong acids and bases, life is easy, right? It's easy because, for example, we only need 2 equations. One of the equations that we need is the ion product of water. Which, of course, is simply that the concentration of protons times the concentration of hydroxide anions must equal 1 times 10 to the minus 14 molar squared. The second equation that we will need is a mass balance of protons. Sometimes called the Proton Condition. Remember that as long as I'm doing a reaction in H2O, this equation, the ion product of water, must be satisfied. So, for example, to give you and again, the reason we can get away with only 2 equations, as you'll see in a moment, is we're going to assume that there's complete dissociation. So let's take an example. We're going to dissolve 1 millimole of hydrochloric acid into 1 liter final volume of H2O. Okay, now this is super simple because you know that that hydrochloric acid will dissociate essentially completely into a proton plus chloride anions, right? And so if our initial concentration of hydrochloric acid is 10 to the minus 3 molar, most of it is going to dissociate completely. So there are 2 ways that we can do this, okay? First, we take equation 1. So what we're going to do here is this is our initial state. And we want to solve for the concentration of protons in the final state. So let's just use our 2 equations. We know, first off, that the concentration of OH minus is simply equal to 1 times 10 to the minus 14 molar squared divided by the concentration of protons. That's our first equation. Okay? And then we know from our second equation that there are 2 sources of protons. We know that H2O must dissociate into a proton plus a OH minus. And we know that hydrochloric acid that we dissolved must dissociate into a proton plus Cl minus. Okay? And so therefore we can write that the concentration of protons must equal the concentration of chloride anions plus the concentration of hydroxide anions. I can simplify this in 2 ways. I can say here that the chloride concentration must equal about 0.001 molar, because after all, the hydrochloric acid is completely dissolved, right? And therefore, I can write that the proton concentration must equal 0.001 molar plus the concentration of OH minus. And if I wanted to, I could use this equation for OH minus so that what I would end up with, I could replace the concentration of OH minus with 1 times 10 to the minus 14 molar squared over the concentration of protons. And if I do that and I plug that in, then I'm going to have a quadratic equation in protons, right? And I could solve the quadratic. Okay. Or I could just make some reasonable assumptions. I can assume, for example, that since it's an acidic solution, right, if it's an acidic solution, then clearly you would agree that the concentration of OH minus is a lot less than 0.001 molar, right? So the concentration of hydroxide anions is a lot less than the concentration of chloride anions. So I can just neglect this, right? So I can therefore say, so the proton concentration, therefore, is going to be approximately equal in this case to the concentration of chloride anions, which is equal to 0.001 molar, right? And therefore, the pH is now 3. So the pH started out for H2O, started out at 7, and then after I added 1 millimolar of hydrochloric acid, it dropped to 3, and I had a change in pH of 4 units. Easy, super easy, if I assume complete dissociation.